Tonight, we have Bill Bengston with us. Bill, I met Bill about 10 years ago, right after his book came out, The Energy Cure. I met him after we had started to do some of our own practice at the Rhine. We have an energy healing team at the Rhine that started out specifically following the protocol that Bill laid out, which he'll talk a little bit about tonight. And we've continued to meet for about 12 years now. Well, actually, more than that now. Uh, but Bill was, from first time I met him, I knew he was doing amazing work. And I think I remember asking him, why doesn't everyone know about this? Well, hopefully, we're going to hear a little bit more about it tonight because his system of healing that he has taught and helped other people to develop through his books and through his audio books has provided it's an advanced system beyond many of the other systems of healing that I've ever learned in my life or that I've heard about. Not only this, but his research is top notch. Bill is a research methodologist who's excellent. He was the president of the Society for Scientific Exploration for, oh, I don't know, it seemed like about 10, 15 years. I'm not sure how many years, but he's like 100 to him. But he's also been publishing in many different journals and has a number of research articles. And you'll learn more about him as we go on tonight. Thanks for being here with us, Bill.
Well, hi, everybody. Thanks for coming. I'm obviously talking about transitioning to healing as conventional. And the general idea here is that although I've been doing research in healing for many, many, many decades, I think 100 is an understatement. I'm trying to turn it into something that's not exotic and rather turn it into something that can be conventionally found and conventionally applied. So we'll look at some research that points in that direction, and we'll see where we go. If you need to contact me, can you see my pointer? Is that working?
Oh yeah, we see that, Bill.
I'm on info at, there's an email you get me at, and if you want to look at a website, bengstonresearch.com. So let me see if I can get this thing to go. I cannot. I can. Okay. John alluded to, he didn't allude to, he said he read The Energy Cure. It's still there. It's still very old, and there'll be a new edition of new stuff that's happened since 2010, which is a whole lot. We're working on that right now. But if you want to read my story and how I got into this and such, The Energy Cure, if you want to learn how to do the stuff that we're talking about here, I don't have real secrets here. I don't have secrets hidden up my sleeve about how to do this stuff. There's a CD set or there's a download or something like that that's called Hands-On Healing. And I think, John, this is how you started, listening to the CDs and then trying to replicate the stuff.
Yeah, we sat as a group and we all listened to the CDs and started trying to learn the process.
Yeah, so there's a lot of different ways to learn it. If you want, you can look on the website. The website will have ways to learn this. It'll have workshops. It'll have people who are practitioners. There's a lot of stuff there. Play as you will. John also alluded to publications. This will be found on the website, and I think I've violated a number of copyright laws by just putting articles up on the website. More accurately, Margaret's put the stuff up on the website. So if anybody comes to arrest anybody, they'll take her away. Um, some publications, I think there's maybe 30 or 35 publications that you can look at. These tend to be on the geeky side. The book and the CDs are kind of straightforward and spoken in normal language. This is where the meat is. And so if you like meat, look at some of the selected publications, and there's page after page after page of this kind of stuff. So let's get into the heart of the matter. I'm going to suggest to you that there are 3 stages of healing research, and that we've been stuck. This is an editorial. We've been stuck on the first stage, which is proof of concept. In other words, is the stuff real? Does healing really happen? You know, it'd be the same kind of thing that parapsychologists do. Do you believe in parapsychology? Do you believe in ESP? And frankly, if I were a parapsychologist and I was asked that question, I would say you're a dope. There's been, as John said, 90 years of research at least going on about this stuff. There's a plethora of publications. There's tightly controlled experiments. There's all sorts of stuff. And the question of do you believe in it, I think is naive. And I think it's dopey. I think you need to move on from that kind of stuff. I think the same is true of healing research. Healing research is unorthodox. And so the initial reaction is, do you believe in unorthodox things? And you approach that with a certain amount of caution. But at this point, there's been many, many, many years of proof of concept on healing is real, including myself. And I'm going to show you some of that stuff, just a quick thing. But then I'm going to go on. My focus tonight will be on storage and scaling. So we can ask, does healing, is it real? We can also ask, can we store it? We can also ask, can we scale it? And I think that if we can answer the questions of storage and scaling, we can convert from stupid questions like, is this real? You really think this is real? Could it be real? Sounds too good to be true, or something along those lines, to making it conventionally available as a therapy. Why not? So let's look at the stages and what I'm talking about here. If you think of healing, this is pretty much the model that you think of. You think of a healer and you think of a healee, and I don't know why their eyes are closed, but they're there and they're putting their hands around, or they're putting their hands on, or it's hands-on healing, et cetera, et cetera. And everybody knows this general idea. And so you think of this, and what I'm going to suggest to you is we've spent a lot of time working on this. Does healing really happen? And a subset of that, what are the correlates to it? But it doesn't address the question of storage and scalability. And that's going to be my focus tonight. So we'll talk a little bit about the concept, but then I want to focus on the storage and scalability. So we're used to storage and scalability in our ordinary life. This, I think, the picture you're looking at is, I mean, it's a ripped off picture, but it's a picture of kind of the classic kind of healing. But if we want to move on from that, how do we put this in a bottle? And we see this in our daily lives, all sorts of places. Imagine being Benjamin Franklin 200 something years ago, trying to, flying his kite, trying to show that lightning, there's electricity and lightning, and playing with lightning and electrostatic stuff. And imagine what Benjamin Franklin would say if he saw a battery. I mean, he'd freak out. Here he is, he's trying to capture something in the sky, thinking that the thing is electricity. We're trying to capture healing in the sky, trying to think it's this, that, or the other thing. How do we put it in a bottle? How do we put it in a box? Well, we can put electricity in a box. And we know about batteries. And we can scale batteries. We can make a boatload of them and pick a battery. I can have a battery factory. Way past anything that Benjamin Franklin would know, comprehend, understand. He'd be flopping on the floor. So let's look at stage one. Is healing for real? Well, I'm going to show you a couple of pictures, and I'm going to make this as short as possible. And this is going to be on selected cancer experiments using mice. And so the way to read this, if my pointer's in the right place, is we're dealing with a mammary cancer. I have 2 formal experiments on mice at the City University of New York, 2 at St. Joseph's College, 1 at Arizona State, 3 at Indiana U, 5 at Brown, et cetera, et cetera. I've studied sarcomas. I've studied oncogenic mice. I've studied nude mice. Don't worry, I have no nude mice pictures. I've studied extremely aggressive. I've studied the effect of recordings on breast cancer, bladder cancer, melanoma, and et cetera, et cetera. The list could go on, but you get the plot. I've used a lot of places, a lot of respectable places, and done traditional research on a non-traditional subject. So short version, mammary cancer. some gobbledygook here, but the thing that matters here is in this particular form of cancer, in this particular form of mouse, you inject the mouse with a certain number of cells and they're all dead in a month. I mean, that's the bottom line. Every researcher all over the country knows about this mouse. There's over 2,000 publications on this mouse. It's traditional stuff. Any oncology laboratory would have this all over the place. And what happens is very simple. You inject, you wait, you try stuff. If you're in biology, you give them vitamin C, you give them this, you give them that, you try to make them last more than a month. And so far, after 2,000 publications, no mouse has ever lived past a month. And they die ugly, and this is ugly research. You don't have to be a biologist. The fur is falling out. If my pointer's in the right place, this is a tumor. The eyes are all messed up. This is essentially a dead mouse. And that's the classic, this is what happens with the mammary cancer. What we do is we put our one model, we put our hands over the cage. We do the cycling technique that I've invented. This is me at Brown University. We wear funny clothes. This is not regarding COVID. This is regarding having a clean lab. And what happens is weird stuff. The tumors get a slit, the tumors ulcerate, and the tumors implode, and the mice are cured. Mice are cured means they're not remitted, they're cured, meaning they live their entire lifespan. They can't get cancer, they don't get cancer again. There's no change to anything except the cancer is completely gone, and they're cured of that which has killed 2,000 experiments at least. So, proof of concept. Is the healing for real? Yes. You do. I've done 20 experiments on cancerous mice. I've done a boatload of experiments on cancer cells. I've done a boatload of experiments on non-cancer things. Yes, healing is real. It doesn't mean all claims are real. It doesn't mean the person who says I'm a healer can really heal. It doesn't mean anything like that. It just means something is out there. My question is, can we capture it? Can we put it in a bottle? Can we make that bottle widely scaled? So let's look at some correlates from stage one. Is healing real? We'll just look at them real quick because it's not my focus. What are some of the correlates to healing? Well, the first one alone, we could spend the entire night on distance. And it turns out distance is irrelevant. Now that's anybody who's involved in healing research and anybody who's involved in parapsychological research will say essentially the same thing. None of this weird stuff degrades with distance. And my point is simple. If none of this stuff degrades with distance, what in heaven's name is it? What else do we know that doesn't degrade with distance? And the answer is, I don't know anything that doesn't degrade with distance. So if I shine a light at you and I start walking backwards, it gets dimmer. If I start yelling at you and you're listening to my voice and I start moving away, eventually you can't hear me. And so we're all familiar with that. And the degradation of any signal is a simple formula, and it's predictable, and it's reliable. In this stuff, distance is irrelevant. It doesn't degrade, which means, in my terms, it's not energy. At least it's not an energy that we recognize. And so energy degrades by a simple formula. Healing does not degrade by that same formula. I could have cancerous mice here. I'm in New York, and you're in, or many of you are in North Carolina. If I heal a cage of mice in North Carolina, I get the same results if I'm here as if the mice were here in front of me. That's something to spend a lot of time thinking about. And I understand the irony because my book's called The Energy Cure, and I don't think there's any energy, but we move on. I don't know what it is, but it's not energy as we understand it. Another, we could spend a night on dose response. There is a dose response. If you give a lot of treatments, you give a little treatments, you give longer treatments, you give shorter treatments, you give. Dose response turns out to be a very, very difficult problem to solve. Again, I can spend a night doing a couple hours on dose response. I'm just putting up here now that I could demonstrate and defend there is a dose response to healing. I could demonstrate to you, but we don't have the time. Healing occurs in bursts. And I'll just show you some bursts because it's kind of cool. Here's a mouse. Here it has an ulceration on the side. And this is 22 days after injection. And I'm going to show you the mouse 6 days later. It's gone. And this is a biologist pulling the fur back, looking for where it was. So it was, and then it's gone. And if you don't pull the fur back, it's like it never had it. And 35 days, this can be stretched out to 2 years. And the 2 years, there'll be no sign that the mouse ever had cancer. That's reasonably interesting. Belief is irrelevant. I don't think the mice believe. At least I've asked them and they seem to ignore me. I don't think cells believe. I don't think that belief is irrelevant. I mean, is relevant in humans. And in fact, in humans, I think belief might work against. That's another night that we could spend talking about this stuff. When you believe something, here's the Cliff Notes. When you believe something, you have a tendency, not you, there is a tendency to defend those beliefs. The tendency to defend beliefs brings this stuff into the conscious mind. I don't think the conscious mind is the source of healing. And so if you impose, you believe this, I don't believe this, and you get into this intellectual argument, it's not helping. It's not helping at all. We could spend a night on placebo effects. And by doing healing research, I've uncovered some, I think, pretty interesting phenomena in placebos. I think I can explain how they work. But I found placebo effects in people. I found them in mice. I found placebo effects in cells. And for a head twister, I found placebos in placebos. I won't go into that now. If you want, we can ask me a question and we'll talk about it.
So Bill, let me just before you get on, because I know you have some other really interesting things to get into, but there's some questions that came up in the chat that I want to ask you so you can address them.
Chat away.
Well, first one is how often does this occur, this healing? I mean, you're doing this with all these different experiments, and you showed one mouse that was healed. How often does this happen?
What's the rate of cure?
Yes.
If we do it straight and we're not doing anything else but healing, that's different than trying different stuff that we're correlating with. So if we do it straight, I'll give you a guess. The full cure, it's not 100%. It's probably in the low to mid 90s.
It's tremendous number of
Yeah,
that's a tremendous
right
effect.
off,
It's a very large effect.
it's a very large effect. Right now, I'm thinking back to the various combinations and permutations. There's one mouse I can't account for in hundreds. So maybe the effect is higher 90s, but it's not 100%. And the one I'm thinking of, which is the one that has kept me up at night, same volunteer healer, because I don't do many of these anymore, but I get non-believing, inexperienced people to just heal. It's not a major deal, but they do my cycling technique. They get a cage, they don't believe it's going to happen, and they do whatever the protocol is. Same healer, 2 mice in the cage, injected on the same day with the same amount of cancer. They're essentially clones of one another, and the only person who's ever seen them is this one healer. One mouse gets cured in X days. The other mouse takes 2.5 days, 2.5X to get cured. So if you ask me how many get cured, the number is very high. If you ask me speed, the standard deviation is large.
Ah, Okay.
And it's also the same anecdotally in people. So the correlates are how many treatments does it take? No way to answer it.
Hmm.
How many, like that. So just how many could we cure if we're trying to cure? I would say at least mid-90s. And why the ones that fail, they really fail. And the mouse don't believe that I can tell. And so we've had a mouse, this one mouse that haunts me, treated. Tumor gets bigger, 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 like you saw on an earlier slide, and it died of cancer. Now, I don't come to that and I say, you should have believed. They had
What
enough
was the
dose.
procedure to do this?
I'm sorry, I didn't catch that.
He was
The,
asking the
the
healing
steps,
procedure.
the healing steps.
I'm not sure I understand. I'm
He,
sorry, I
he,
don't mean to be
he,
annoying, but I don't understand this, what you're asking about steps.
so he's asking about the procedure you use for the healing.
Oh, okay. It's a technique called cycling. It's contained in the CD set that John learned and a bunch of people down there at the Rhine learned and are reproducing some of the stuff by using it. It takes more than a quick answer here. John, would you say that's fair? It takes more than a quick
Oh,
minute and a
oh
half
yeah.
answer. If
It took us 6 months to develop it, and then we realized we still didn't know what the hell we were doing at that
Yeah,
point.
and if you're taking a workshop that we offer to teach people how to do this, it takes a couple of days. So I'm not trying to be difficult, um, but this stuff is published and it's public, and so I'd be happy to communicate with you offline, but I can't make this a teaching moment,
Right.
meaning a teaching of what the techniques are.
So, but getting back to the mice, you said when you treated them, then they were cured. Does that mean that they can't get cancer again?
Cured, the parlance for cure is did it go away and it's gone and it's not coming back? But being cured of X doesn't mean you're cured of Y. We haven't done those experiments. So we've taken, and we'll see that in a bit, we've taken mice that have gone to cure, meaning there's no sign of anything cancerous in it, and we've re-injected them. That cancer won't take. The mice not only are cured, they're immune to that, at least that kind of cancer. The next follow-up question would be, are they immune to other types of cancer? I would predict no, but the reality is we haven't tested that yet.
Okay. Well, so I only have one more question I'm going to ask you before I let you get back to it. You say there's a dose response,
Yeah.
meaning, a higher level of treatment can have a different effect than a lower level of treatment. Have you noticed,
It's not that simple. I don't think it's linear.
okay, well, have you noticed an overdose
No.
response? Is it possible to get too much of this treatment?
No. We've done well, I mean I can't give you an upper limit that anybody's ever tried, but I would say that certainly there are people who've been treated virtually daily for many, many months. A mouse experiment, if the mice don't turn around in a month, they're going to be dead. And so we've tried an hour dose per treatment, a half-hour dose, 45 minutes per dose. We've tried treating 5 mice at a time, 10 mice at a time. We've tried large numbers of mice in an experiment, small numbers of mice in an experiment, looking to see what are the correlates and how do they relate even as an inverse to a dose response. And so I can say with some confidence, there is a dose response because we've pulled it back, pulled it back, pulled it back, pulled it back. And then you start to get a wobble in the model. I don't know what the minimum is, but I'm guessing there is a minimum out there someplace.
But you haven't ever seen anything where you exceed a safe amount of healing treatment.
No,
Great. All right,
no.
Bill, I'm going to let you get back to your presentation, and so I'm going to jump out of here again.
Okay, and jump in whenever you think it's appropriate to jump in. This doesn't have to be a formal event.
I'm not a shy man, Bill.
Really. Uh, so if you want to spend a really good night, we come in here and talk about placebos, and I'll show you why it's not psychological. It's rather an actual physical bonding between spatially separate groups, but I digress. So more research correlates, just a few more. Healing is stimulated by the needs of the healee. I don't think this is PK. And so I understand I'm speaking to a parapsychological organization, but I don't think healing and PK are strongly related. They may be second cousins twice removed, but they're not the same thing. Depending on what the healee needs is what's going to go on. And different healees having different needs. It's not me imposing my will on mice or people or anything like that. I could defend, but I'm not going to try to tonight, that healer and healee go into resonance. They bond. And they bond even if I'm here and you're in North Carolina. I've done brain research using EEGs. I've done brain research using functional MRIs. I've done EKGs. I've done. There's a resonance which goes on when healing takes place, and it's real. It's reliable. It's reasonably interesting. Healing also changes the physical space that healing happens. This could be another night. Something happens when healing happens, and it lingers in the room. And I can show you some geeky things, and actually I could show you a lot of geeky things about some frequencies and such that we have, and we get some really anomalous output on geomagnetometers and all that kind of stuff. And it happens in the room even after the experiment is over. Healing is not a field effect. I don't put out healing and anything in my benevolent path is fixed. There's not a field effect. There's much more specificity of target. So let's say, for example, I'm in New York, you're in North Carolina, you have a cage of mice. I'm in New York, you have a cage of mice in North Carolina. I don't put out a healing wave that I hit the target in North Carolina. Now, again, think about that. I hit the target in North Carolina. There can be a second cage that I'm not going to hit, and those mice die, and the ones I hit, those mice live. If it's a field effect, you wouldn't have these type of phenomena. If it was really a field effect, if I'm in New York and you're in North Carolina, everything between me and North Carolina would be getting a treatment. That's clearly not the case. And we can go on, but it's not my topic for tonight. So these are some of the correlates that I could defend. Here's some stuff I don't have any idea about. I mean, I don't know. Is healing compatible with conventional treatment? Well, it appears maybe, sort of, kind of. For some things, it's more compatible than others, I think. But the reality is we don't really know. So if you take a medicine, you take a conventional medication, and you do too much of it, this goes to John's question before, there's a consequence. You take too little, there's a consequence. You take too much, there's a consequence. I think John's question was coming from a similar place. Does this happen in healing? Too little, I think, doesn't happen. Too much is a waste. It doesn't respond like conventional treatment, but is conventional treatment compatible with the healing? Should we do them sequentially? Should we do it as an alternative? Should we do it as complementary? We just don't know this stuff. It's doable, it's researchable, but you only have so many times that you can do research in so many minutes and so many hours and so many days. Is healing energy? I don't think so. Is it information? If it's information, that doesn't help me understand how it works. How do distant treatments hit a target? Different treatment techniques, do they produce different outcome? How much of the healing depends on the individual healer? How much of the healing depends on the healee? Can we make a chargeometer? Can we make a healometer? What's the effect of multiple healers? What's the effect of multiple healees? how much of this is teachable, and I could go on and on and on. So what we don't know vastly exceeds what we do know. And what we don't know that's the most confusing is we don't know the things that we don't know to ask. This stuff doesn't make sense by our ordinary understanding of energy, the way the world works. It's different. So let's be different and let's look down the road and say, so the stuff is, whether we understand it or not, can we capture it? Whether we understand it or not, can we capture it and scale it? And the answer is yes, we can capture it, think battery. Yes, we can scale it, think battery. So let's talk about work on storage, and then I'll talk about work on scaling, and then overlaps here too. So I've spent, I don't know, a couple decades working on storage and scaling, and I have a bunch of experiments indicating that healing, and you can call it healing intention, can be stored, and it can be stored in a variety of organic material. It can be stored in a variety of inorganic material. So examples of organic material, for example, cotton. And by organic cotton, I don't mean the kind of stuff that you know, like I want to have organic food. I want to have organic cotton. I want to have organic whatever. I mean the classical definition of organic. You got a base of a carbon atom. So I've done some experiments on organic cotton, cell medium, treated mice, to name just a few. Inorganic material, water, recorded media, using a variety of recorded media techniques. And we'll look at that very quickly. So here's an ordinary piece of cotton. It's an organic piece of cotton. All cotton is organic, whether it's been pesticided or not. So we have cotton as an organic target. Can we treat cotton? That's the question. So I don't have a piece of cotton with me, but can we treat cotton? In other words, could I take this piece of cotton, put it in my hand, put it in my hands, whatever technique I have, put it in my hands, and do the healing intention stuff? Will my intention work its way into the cotton somehow? And I don't really know whether work its way into is the right way to think about this, but if I treat cotton as cotton, can healing be produced? And the answer, of course, you're probably not going to be surprised, is yes. And we take cancer cells, and this stuff we did at Brown University, we take cancer cells in a dish, well plate, we take cancer cells in a dish, and we have treated cotton and we have untreated cotton. If the cancer cells start to get near the treated cotton, they start to genomically flip. If the cancer cells get near untreated cotton, they don't do anything. But if a crude way of saying this is cotton can affect cancer cells, the inverse, cancer cells can recognize when cotton has been treated. And we get this protein flips, that protein flips, this p-value happens, that change. Healing can be stored in cotton. We've done a bunch of, and I'm doing this quick, I understand. We've done experiments with cell medium. Cell medium is the liquid that's put in the well plates, the containers that you grow cells in. And this is just standard off-the-shelf stuff. Any lab in the country is going to have this stuff sitting on the shelf. We get the stuff, we treat the cell medium, stick it in the mail, and let another lab say, what happens to the stuff you use this on? So we take this, the cell medium, do our treatment, ship it away, and then we throw some cancer cells into the cell medium that's been treated. And what happens is pretty interesting, and it startled some people. If the cancer is grown in regular cell medium, we'll call that the control, we get a certain response. If the cancer cells are treated with treated cell medium. It varies a little bit, but you get up to a 600% increase in cell growth. The cancer divides increased by 600%. Now, some people get upset with that. What do you mean your cancer is dividing? I thought you were supposed to get rid of cancer. If your cancer is in a well plate, in a container, in a dish, in a flask, and your client, using that loosely, your client is cancer, and you give it jazzed up cell medium, it gets all excited and uses the stuff to do what it likes to do. Cancer cells like to reproduce. If we give them healing to a cancer cell is reproduction. And so what we found is about an average, there's a variation, but about an average of a 600% increase in cancer cells. I think this is speculation. If we take this jazzed up cancer cell in vitro, we take the cancer and we stick it into a mouse, the cancer will implode. I'm not saying we've done that. I'm saying this is my hypothesis. I think it would implode because I think healing, still driven by the healee, goes to the higher order need of the organism. So if I'm treating a cancer cell, the need of the healee is give me stuff that'll help me grow. If I'm a mouse that has cancer, give me stuff that'll help me get rid of the cancer. Either one will work. You don't have to know which one you're doing, because frankly, I don't think healing has much to do with the attitude and beliefs and such of the healer, nor the healee. Organic. Over here, we have a treated mouse. When it's been in the, I mean, it's actually ripped off the internet. That's a rat. That's not a mouse, but I couldn't find a good mouse. Treated ulcerated mouse, like you saw in an earlier slide, and you have an untreated mouse with a tumor. A mouse that's been treated, it's going to ulcerate and it's going to implode and the mouse will be cured. The untreated mouse, the tumor will grow and it will grow and it will kill the mouse. If we take a treated mouse, one that's been exposed to the healing technique, and we do a transfer of fluid, and the fluid can be blood, the fluid can be tissue, and give it to an untreated mouse, that mouse will be cured. Now, what does that mean? Well, it means it's weird, but the second thing it means is this mouse that was treated contains within it the information, I'm speculating, contains within it the information to fix this mouse. So if a mouse can fix a mouse, which can fix a mouse, which can fix a mouse, we're starting to see then hints of scalability. We just don't want to go around injecting a whole bunch of mice. Inorganic material, water, good old ordinary water. If I take good old ordinary water, I have a glass of ordinary water here, and I hold that water and I do the cycling technique, which is what was the earlier question was. If I do the cycling technique and hold this for some unknown number of minutes, it's probably a dose response. And I take that water and I put it in the mouse cage water bottles, and I never meet those mice. Those mice will be cured. In other words, stated simply, the water now has the information that can transfer. If we can treat large amounts of water and give out that water, a hint at scalability, but the scalability is low. It's scalable in the sense that maybe water can treat water, but we got to test that. Does cotton treat cotton? All I can say is But you see the idea, if cotton could treat cotton, it's scalable. Cotton can store healing intention, Water can store healing intention. Lab medium can store healing intention. A mouse that's been cured is storing healing intention. So we get a nice, cute little XY graph here. On the x-axis here, scalability. Is the scalability low? Is it medium? Is it high? We, of course, want it to be high. Effectiveness. Is it low, medium, or high? So we got hands-on healing and distant healing. It's the same thing. We do hands-on healing and distant healing. It's very effective. So again, we got a zillion experiments. Yes, it's effective, but it's not scalable. So you come up, you put your hands around a cage, and that cage is fixed. Put your hands around a person, and that person is fixed. What if there's 100 people? What if there's 1,000 people? What if there's 1,000,000 people? What if there's 10 million people? It's not scalable. Even if I teach people how to do hands-on and distant healing, and they teach other people to do it, and those people who've been taught, and we're down generations, the next generation, you still, how many people do you have? As distinct from how many people need treatment? And if you think about it for more than 4 or 5 seconds, there's an inexhaustible supply of people in pain. So maybe it's a little scalable, but it's not a lot. So this up here, which works, there's a limited amount. The storage scalability thing, over here, cotton, treated water. I probably should move it over to the left a little bit. Cotton is storable, but I don't really know how to scale cotton. Treated water, which is water that you've actually held and treated with a focus on intention on this water, unless water can treat water, it's not scalable. So where do you want to be? You want to be in the top right-hand corner. Top right-hand corner means maximally scalable, left, high effectiveness. So over here, what I'm calling informed water is water that I haven't talked about yet, water that can reproduce itself and contains within it healing intention, and that healing intention and that water can be scaled. But the place you want to be is over here in the top right, maximally scalable, maximally effective. We've done a bunch of different types of recordings, which I'll talk about briefly here, but it's not my focus, on recording healing. And it turns out, if you think about that, recording healing is maximally scalable. In fact, the informed water should move over there, and all this stuff should move over there. And the recording, if as good as hands-on, would be maximally scalable, meaning I could take a recording of healing intention, whatever that means. I could upload it into the web. I could make it globally scalable. You can't get more globally scalable than a recording. So scalability, the recording is going to win. Effectiveness, this recording is not as good so far as informed water. We've done recordings in ordinary studios, sound studios, and I don't think it's as good as a fancy recording we made, and I don't think either of those is as good as informed water. Now, the good news is we've created a technology in recordings and water that we can externalize from us, and we can build upon like any good technology. So I build the first Model T. I'm not finished with my car research. Everybody can put their 2 cents in. Different car companies can work on that, and we can raise the effectiveness of the car. We can scale the car. We can do all those things once it's a technology. And what I'm suggesting to you is I have a technology for the recordings, and I have a technology for the informed water, but so far the recording's not as good as the water. Treated water means here's the water I've actually treated. Informed water is water produced by a device. that can be scaled. We don't need to be sitting there with our hands around the water and doing all that kind of stuff. So the storing, scaling, healing research arms, we're shooting for the top right corner of this slide. We'd like to have maximum scalability and maximum effectiveness. And so I have formal research going on with recordings, with a Faraday cage in studios, sound studios, and I have a research arm that's dedicated to water. I've done the treated water to my satisfaction. Really, I'm doing now primarily informed water, which is water that has been mechanically, using that loosely, mechanically produced. Let's start with the left-hand side and end on the right-hand side. So a recording, I'm going to talk here about where the recording takes place. We have recordings that take place in a Faraday cage and recordings that take place in a studio, a sound studio. We have basic research on recordings. We have clinical research on the recordings. So let's look at the recording. This is the inside of a large Faraday cage. Faraday cage is screens out electromagnetic, not all, but it screens out electromagnetic signals. It has limits in terms of how low, in particular, how low it'll go. But this is a 12-foot by 12-foot Faraday cage, which has it's built specially to be electromagnetically clean. And I just need to tell you that if you've never been in a Faraday cage and spent some time in there, I strongly recommend it. It's quite an experience. All of us now are living in electronic soup. We know we're in electronic soup. We're on Zoom, for crying out loud. We're sitting in front of a computer screen, for crying out loud. There's stuff in the air. We're living in electronic soup. This screens out this soup. And so we got a 12 by 12-foot Faraday cage, and we put in there a bunch of stuff, a bunch of detectors. We actually put in there 33 different types of detectors. We take, oh, let me go back. We put 3 people, I won't mention who they are, 3 people in this cage, holding a piece of cotton, doing my healing technique. And I'll out at least myself, I'm one of them. So there's 3 people who are experienced with cotton and treatment and all that kind of stuff, treating cotton in a Faraday cage while 33 detectors are detecting stuff. Some of the stuff it's detecting, we don't even know what we're doing, but we're throwing a kitchen sink at it. So a caduceus antenna, that's somewhat speculative. Depending on who you talk to, you'll say that scalar fields are established. Some will say they're not established. They're hypothetical. They're theoretical. But we measured. We got detectors looking for scalar fields. We got stuff for electromagnetic antenna. We've got magnetometers. We've got in there. 3 people go inside the cage, holding a piece of cotton. We're running the detectors. 33 detectors are detecting stuff that detect those 33 recordings are now crushed into a single wave file, which can be played as an ordinary audio. That audio is played to cancer cells in incubators. So we have an amplifier, we have an incubator, we have cancer cells, we have speakers, and the same way you'd play a CD, we're playing the recording of the 33 detectors reduced to a wave file to the cancer cells, and we want to see what happens to the cancer cells. So this is a pretty elaborate setup, frankly. The funny part is the amplifier is cheap and from Amazon. The speakers are cheap and from Amazon. We bought them because they fit in the incubator. The production of cancer cells is a little more expensive. The recording is in 6 figures. It's an expensive stuff, but we actually play it on $49 speakers and a $49 amplifier. And I'll come back to that later, maybe. So what happens? Well, this is actually a publication that you can get on, I have on reserve. in my website. But this is a little bit of a breakthrough because this is published in a traditional biology journal. What happens when you play this WAV file with 33 recordings compressed to cancer cells in an incubator? And what happens is pretty interesting. We looked at 167 genes related to cancer, and 68 of them, this is done at Brown, and 68 of them genomically change when they hear that they don't hear, when they're exposed to this recording. 167 genes as targets, what happens? 68 of them reliably change. Now, because they change, and they change so dramatically, we didn't believe it. I mean, I don't believe this stuff. So we didn't believe it. So we replicated it. We replicated it. We replicated it. We replicated it. We did it 7 times. And we get the 68 genes. So we can conclude, and did conclude, reproducible biologic changes have been induced by healing intention. Storage. Healing intention can be captured and released, thereby allowing scalability. Hands-on healing, we did some A-B comparisons. Hands-on healing on the same cancer cells, hands-on healing produces a stronger response than the recording as presently constructed. But it's a technology, so it can be, we hope, improved. And right now we have experiments going on at Brown with what we hope is an improved version of the recording. Again, we're looking for the next car model. We're looking for the next gizmo. We're looking for the cars to drive themselves. We're looking for the recording to get stronger. We're looking for the recording to get as good as hands-on. Right now, it's not as good. It's getting close, but it's not as good. If you needed healing help and door number 1 was a recording and door number 2 was hands-on, I'd still take hands-on. The recording has the potential to equal or surpass hands-on, but we're not there yet. And we did not put into the article, healing activity produces a conditioned space. We thought that was we found that, we tested that repeatedly. And the short version is this: once the incubator has been exposed to the recording, the incubator is now, I don't know the right term, quote, don't take it too seriously, charged. So without a recording on, we can stick cancer cells in that. We tried it up to 3 days later, and the biological changes still change. We didn't put that in the article because it's too crazy for most biologists.
Hey, Bill, I feel like I need to jump in here because there's something that's really not clear. And I don't know if I don't know if it's clear to you either, but what was the rationale behind taking all of these different readings and all of these different this different data that was collected and turning it into a recording? Why did you do that?
We did the recording because it's easy to apply. Nothing more exotic than that. And the reason for all the different detectors was because we don't know what we're doing. And so it's a kitchen sink approach. So we got the caduceus thing and the electromagnetic thing and the magnet thing and and they're all going, I hope we capture something. So it's the 33 detectors is because we didn't know what detectors to use, and we still don't know whether those 33 detectors are right and we can improve on them, but are they the ones that we actually settle with?
But
And here we're arguing essentially like, give me the characteristics of your battery versus my battery. And I use a different technique to do this kind of battery. You use the other technique to produce that kind of battery. Both of us can make it better. And I come along and say, what are you doing that for? Well, if I combine yours with this and I combine this with that, so it's a kitchen sink. We're throwing a soup of all of that stuff. It turned out to be a complex problem, and we had to actually build a water process supercomputer to take the amount of information, reduce it to a WAV file. Is there a better way? I'm sure there is.
Well,
It's like I have a
the
model,
question
I have
is,
a Model T, you have a Model G. We can both improve.
so you have all of this information or all this data that's been going on that you're collecting, and I
Yep.
understand the idea of having a kind of kitchen sink. We don't know what's working. Let's try it all.
Right.
But why an audio file instead of a video or instead of something else? Why is it an audio file?
It, it's easy to use. It's easy to do. We can do the kind of stuff we were hoping to do, like stick it in an incubator and find out in vivo, in vitro, and all the other VOs. So it's easy to do. It's transportable. It's nothing more than convenience. And frankly, a good chunk. I have a suspicion. As we as we did get together and yell at each other about what kind of detection devices we would use, nobody agreed. So it ended up being a kitchen sink. And the whole thing may be wrong, doing something, but not particularly Model T, current fancy car, light years apart. And that may end up, hopefully, if the technology gets better, I can build a better battery. You can build a better battery. And, hopefully in a couple of years, we'll have the Teslas going 500 miles between charges. Why? Because they got a better battery. They're doing it in a different way. Same idea. This is step one. This isn't the final project.
So I guess another way to ask the question, I understand your answer. It's that this is what worked, but I'm wondering if are there other delivery mechanisms? I understand collecting the data and turning it into something, but have you tried other delivery mechanisms?
Oh yeah, I'm getting
Like
there.
light or other things? Or
Not light. Only recently have we asked the question, can we turn this into a light phenomenon? But so far we have not.
okay,
So
well,
we're working on juicing up parts of the recording and parts of the output and seeing if we can, turn the dials and make this part stronger and this part weaker on the parts that we find interesting. But we're testing actually right now. We're testing using incubators and such. It's going on as we speak. But we don't know whether we're pulling the right dials.
right.
Yeah, so again, first car, first battery, first attempt at this, and it's holy mackerel, something's there, and it's a technological question, which is wonderful as opposed to its hocus pocus, and things like that. This is done with ordinary equipment. It's a little over the top, but it's done with ordinary equipment that people would recognize.
Well, I'll let you continue with the presentation. It's just I felt like I had to jump
Oh
in.
yeah, by all means,
Question.
jump in and do it. And no matter what you ask, I have no qualms about going, I don't know.
Right.
And what this should be showing is, holy mackerel, this is really interesting. On the other hand, I don't think he really knows what he's talking about. And the answer is no, I don't. I'm building the first Model T and wondering if there's a better way.
But if it's working, I think that's where you're going to get next, aren't you?
Well, it's working, so this paper showed it works in vitro. This paper showed that it works in vivo. So this is also at Brown. Instead of being in an incubator, we're in a mouse room, and we have cages of mice, cages of mice, cages of mice, same speakers, but there's no incubator, same amplifier, and it's just being played in a loop. And what happens to the cancerous mice? So we're starting most basic in vitro, what happens to the cells? Now what happens to the mice? And then you can see you just keep on with the research agenda. What else can you affect? So what happens to the mice is we have reproducible biological effects. And we don't care. But let's just look at the bottom. The spleen weight on these mice increased 5-fold. Spleen weight is an indirect measurement of immunology. And something is being stimulated in the mice that is certainly not normal. Now, what happened is Brown got chicken after a while. We were getting ulcerations and we were getting pretty interesting things. I don't have those pictures here. tonight. But they said, no, this is too nutty. The tumors are too big, and they're flopping on the floor. And so in the name of being humane, they killed our mice. That I found rude, but we had a bunch of stuff that we could salvage from this experiment because it was going on long enough that interesting things were going on, and we could publish the interesting things that were actually going on. It showed we can do this to mice. It shows that there's a strong possibility that some mice will be cured. I don't know about it if it's all mice. And there's also a reasonable suspicion we have that this recording, as I showed you on the XY graph before, is not as good as hands-on. It's going down the same pathway, and we're actually testing that now and looking at similarity of gene expression, but it's going down the pathway. Something's going on. I don't think it's as good as hands-on yet. So is this scalable? Yes. Is this storable? Yes. Have I captured everything there is?
Can I ask
No.
a question?
I heard
Alex, if
something.
you put your question in the chat, or you can hold it until we get to the Q&A at the end.
Well, I would forget, this is like very interesting. I'm electrical engineer and I worked on electromagnetic waves most of my life. You're saying here magnetic signals derived from a healing technique.
I'm sorry, I lost what you just said.
How could you prove this?
John, can you repeat what he said? Because I couldn't catch it.
So as an electrical engineer, he's asking, how can you demonstrate that these electrical signals are being captured from a healing technique? The electrical signals that you're using to create these recordings.
Yeah.
I probably, well, I have many detailed analyses of the recording by an electrical engineer, PhD in the stuff, and signal analyst and all that. And so I don't know that I can prove it, but I can say that looking at this as an ordinary, meaning conventional electrical engineer, they're looking at signal stuff and deviations and looking for anomalies in the signal. And there are reproducible anomalies in the signal, which at least the electrical engineer says, what in heaven's name is this? I can't prove it that's directly due to a healing technique, but I can tell you temporally, when you do the healing technique, these things happen.
So I'm familiar with how this process was done. So let me try to explain it in a way. There was an electrical engineer involved in this process.
Yes.
And
Okay.
so recording all of this data, all of the data was taken by the electrical engineer and processed in order to create the recording. That process, what he did, I'm sure it's way over my head. And so, but it came directly from the original data that was collected while healing was being, while you were providing healing, Bill, is my correct?
We were treating cotton. If we do the exact same thing and we don't treat cotton, the phenomena doesn't occur. Those are the parts I'm skipping over for lack of time. So if we just run this stuff and we run the detectors in the Faraday cage, you get ordinary stuff. If you stand inside the Faraday cage, you get ordinary stuff. If you start cycling and you hold a piece of cotton, non-ordinary stuff occurs.
So when you have these recordings and you play them back, what do they sound like? Is it like the
They don't sound
Ramones,
like anything.
or is it like, it doesn't sound like anything?
No, there's no sound that you can hear. And the interesting, from an electrical engineering point of view, I'm not an electrical engineer. And like you, John, a lot of it just goes over my head. So I have literally 75 pages of technical output and analysis. And at the end of that, I just curled up and went to sleep, because I was flopping on the floor. I have to call the guy, 15 times. What are you saying? Give me subtitles. So we have all that stuff.
Great. Bill, I'm going to let you get back to your presentation because I know you got a lot more to cover.
Okay. You said I had to be done by Monday?
Oh, yeah. You can talk as long as you want. I'm here with you, Bill. I'm
No.
leaving on Tuesday, so it has
Oh,
to be before
okay.
that.
Gotcha,
All right.
gotcha, gotcha. So we're trying to treat this as a technology externalized that we can putz with, and we changed. Oh, so the part you asked about hearing, what does it sound like? It doesn't sound like anything. There's a background hiss that's not part of the recording itself. It's kind of like we didn't want to edit any noise out of it. Um, the well, let me just keep going. We tried to take the interesting parts of the recording based on a detailed analysis of the signals. And we don't even know if signals is the right way to do this. So I'm saying it's kitchen sink time. And we found interesting anomalies in the signal at very low frequencies. And by very low frequencies, I'm talking 3.5 to 5 hertz. You can't hear 3.5 to 5 hertz. And it gets more nutty than that. You can't hear 3.5 to 5 hertz. I mean, we don't care what a cell can hear. We don't care what a mouse can hear. Maybe we care what a mouse can hear. But we can't pick up 3.5 to 5 hertz. And in order to play 3.5 to 5 hertz, we can't have little $49 speakers from Amazon. We would need a monstrous speaker to be able to transmit 3.5 to 5 hertz. We would need a very different kind of amplifier to be able to transmit 3.5 to 5 hertz. So, and this goes to the question I was just raising. What is it that you're doing that is captured by the amplifier? And what is it that you're doing that's captured by this, that's transmitted by the speakers? The answer is none of it makes sense. So I'm here as the first or second or third to acknowledge this. What I'm saying here doesn't make any sense. I'm just saying it seems to be stored and it seems to be scalable, but it doesn't make sense. The speakers shouldn't be able to transmit anything remotely like 3.5 to 5 hertz. The amplifier shouldn't be able to transmit anything remotely like 3.5 to 5 hertz. We're finding the interesting frequencies at 3.5 to 5 hertz. So now we're boosting that 3.5 to 5 hertz part of the signal and downplaying the others. So this current genomic work is looking at a modified version of the recording with an estimation of the very low frequencies are being boosted about 100 times. And we'll see if that has any effect on the cells. We'll see if it has any effect, on the organisms, on like that. So we're just trucking along, playing around, trying to figure out how to reproduce this, how to make it better, how to turn a Model T into a modern car with bells and whistles that we can't even think of. And we'll probably, hopefully, look back on a year or so from now and go, boy, were we dumb. I mean, it was sitting in front of us. We couldn't see it.
What is the source of the low frequency?
The source,
Yeah,
I don't know.
but you were able to measure it.
We're able to measure it. There are physical devices that measure frequency way down. And if you're a crazy signal analyst, you got all those gizmos to play with. I don't have the gizmos, but I have on my team people who know the gizmos.
Interesting.
It's more than interesting. It's not, it doesn't make any sense. If I'm making sense to you, I'm not saying it clearly.
Not to me.
So if we go to the other side and we look at water, treated and informed, and we've done basic research in vivo on mice. And this was me telling you that I would hold water and we'd feed it to mice in their ordinary water bottle, in their ordinary cages. And we're doing a study right now using informed water on worms. And I'll get to that in a little bit. If it's in red, it's either ongoing or it's on the drawing board. If it's in dark print, it's complete. We have a bunch of things completed. So we have basic research, we have clinical research, and primarily on informed water. Informed water is the stuff I'm saying takes water that has been infused with healing intention, and that requires so far a human. So you walk around, you take water, you do the treatment like you would have done the cotton, you do the treatment like you would have done a mouse, you do the treatment like you would have done on a person, and you stick it in a gizmo, and the gizmo spits out more water, and the spitting out of more water reproduces the effect. I'll go out on a speculative limb here for a second. I have a suspicion, I can't prove this yet, I have a suspicion that the informed water spit out by a gizmo is actually stronger than the original treated water. We're going to test that, but I don't have that yet. So let's look at water as the target of something you can store and something you can scale. Now, as far as I know, water doesn't have a strong scalability in the sense of a couple ounces of water, I don't think could fix the Pacific Ocean. I can't scale like that with water. I don't know how to do that yet, but it's a technology. So we've got informed water spit out by a gizmo, for which we've done clinical research. We did 3 human trials on taking treated water, putting it in a gizmo. We've done clinical trials, even more tightly done, on people going into a hospital for COVID. We're supposed to be doing a study right now on hepatitis B, a clinical trial, but I think this is going to have to be taken off because the data procedures were violated, and I'm kind of a stickler for making sure it's done right. I want to do a study on long COVID. If anybody has access to a bunch of people who want to do this, I'd be real interested. And I'd be also interested in an antibacterial model, because we seem to be able to do viruses. We haven't tested bacteria yet. But we get informed water. So once again, informed water has been treated by a human. It's given up by that human and put into a gizmo that I helped to develop. I'm not the only one. I helped to develop, and we can spit out a whole bunch of water that has the healing properties in it. It started out healing water, and then it takes on wider properties. We actually have this available if you want any. We have something that is alleged to increase athletic performance. So weightlifters are lifting weights and runners are running faster. This would be an interesting thing to take the Olympics. There's all these doping scandals. I don't think you could be charged with doping scandals if you're taking water. These bottles are filled with water. Restore is a general health booster. Recover seems to target viruses, and Perform seems to enhance athletic performance. And we're going to see add a bottle, add a bottle, add a bottle. We got to see what this thing can do. So now we're talking water, not recordings. And if we're talking water, we can put out a lot of water, but we can't match the scalability of a recording. So recording becomes globally scalable. We can do these drops or these water samples. We can easily make enough for 100,000 doses, but we're not going to get global scalability, not yet. We could get there, but we don't have the stuff built yet to get there to that level. So on Perform, we randomly and blindly given to athletes. They're reporting and actually measuring increased strength for weightlifters, the faster recovery from people doing workouts. We've measured oxygen intake and capacity and all this kind of stuff. And Perform does something. It's going to be the same question as, what does this have to do with healing? And what is it about the water that's different? I'll get to the water in a second. But the answer is going to be, I don't know. I don't know. Jigger it around. Let's see what happens to the battery. Jigger it around. Let's see what happens to the car. Jigger it around. Let's see what happens to the recording. It's a little bit of the blind finding their way out of an alley. And we have a publication on that, on energized water, on Restore. This is Restore produced by a device, not a human. We did 3 clinical trials as pre-clinical trials before we did more controlled. We did 100 people in the States. We did 200 people in the States, 2 separate clinical trials. And we did 100 people in Europe. And incidentally, I don't believe any of this stuff, but 98% of these roughly 300 people from taking this one Restore, and this is drops under the tongue, 2 drops under the tongue 6 times a day. And we measure them for 8 weeks. Every 2 weeks, they're doing all sorts of measurements and they're writing this stuff down and they're measuring this and measuring that. 98% of them report improvements in health. That seems too high to me, but that's what the numbers indicate. Significant increases in physical well-being, mental well-being, spiritual well-being, as determined by the people taking the drops. In a wide variety of conditions, they report changes, including cancer, arthritis, depression, anxiety, pain, Parkinson's, etc. Now, that does not mean all of these were cured. We've had, for example, tremendous difficulty with Parkinson's, but we've had some effects that are really strong, and I don't understand that. Why the variation in responses? The biologists who work with me have said Parkinson's is unlikely to be a single disease. And it's possible, this is speculation, it's possible that our drops work on some variants of Parkinson and not so much on other variants of Parkinson's. Some effects, though, for example, Parkinson's, appear to be stronger than hands-on treatment. We've had, for example, very limited success with hands-on treatments of Parkinson's patients informally in the clinical world. On the other hand, we've had people take the drops of Restore under their tongue. We've had a Parkinson patient who has been in a wheelchair for 5 years, get up out of the wheelchair, get rid of it, and walks a mile a day. I don't get that. And this is from water. Hands-on hasn't been able to reproduce that. So we've got potential in the water that may equal or exceed hands-on. We just have a problem of scalability. With the water, we're using Restore, General Health, Recover for viruses, and a COVID clinical trial, which was double-blind, randomized, placebo-controlled on 160 people going into a hospital to be treated for COVID. So they got the PCR tests, they got multiple PCR tests, they've got this, they've got that, they've got the other. And this is a double-blind study. The physicians giving drops to the patients, the nurses giving drops to the patients, and the patients. Nobody in the hospital knew what they were getting. Some of them were control, nothing, water, and some of them were water, but they were water coming out of this device. So all the water that was used as treatment was informed, meaning it was not laying on of hands kind of stuff. And
What is the
results are-
difference between the waters?
I'm sorry, I didn't catch that.
The difference between the 2 different type of water, the normal water and the healing water.
Yes,
How are they different, Bill?
I'm getting there.
Thank you.
I'm going too slow.
No,
So we did this.
You're not going too slow. We just have an intelligent audience that's thinking ahead and anticipating where you're going.
I like that. Well, the bottom line place I'm going is, I don't know. So I have no problem. I'm not shy about saying, I don't know. You know, how does the recording actually work? I don't know. If I use different detectors, what would happen? I don't know. I got to just test it, and I'm doing this atheoretically largely. So in the hospital study of at least COVID people. So these aren't ordinary COVID people who have sniffles. These are people going into a hospital because they're seriously ill. The control in this is people taking medical stuff and water, not informed water. So they're taking whatever medical treatment they have. We're not asking people to give up their medical treatment. They're taking medical treatment and they're taking ordinary water. They don't know that they're taking ordinary water. The other group of about 80 is taking medical treatment and they're taking our drops. But it's really blinded, randomized, placebo-controlled. We found significant differences between treated and control groups on general feeling of health, temperature, sore throats, coughs, PCR positivity. So for example, people by now are used to PCR tests. You come in, before you come into the hospital, you're taking a PCR test. Once you're in the hospital, you're taking a PCR test. You're taking regular PCR tests. And the people who took our water, the PCR tests go negative after about 2 or 3 days. The untreated ones, or the ones just taking ordinary water, don't have that kind of response. So we found on certain measurements, general feeling, temperature, sore throats, coughs, PCR, very significant differences between the groups that were treated with the charged up water coming out of a machine versus controls. There were no significant differences, no difference between treated and control groups on headaches, fatigues, aches, eyes, shortness of breath. And these tended to be things which are short-lived. So you come in, you have a headache, whether you take my water or not, the headache's going to be gone pretty soon. And therefore, there's not going to be much difference between taking treated water and untreated water. And you can see this on just, if we graph this out in various ways, we'll see you're coming along and pretty quickly, the people taking the treated water, there's going to be a difference between them and the ones taking untreated water on a whole bunch of different measures. Everybody's getting medical treatment because, again, we're not stopping that. But if you take the treated water, even if you don't know what you're taking, it makes a big difference. The difference is stronger than medical intervention. Now, can you imagine? Saying out loud, water produces a stronger effect than does medical intervention. And I'm telling you, water produces a stronger effect than medical intervention. This became, I think, a pretty interesting article. Double-blind, placebo-controlled clinical trial hospital using informed water. This is also on the website. By all means, take a look at the geeky parts. And now, if you go to John's super audience, what's in the water? I would give John's right arm to know what's in the water. We're just doing that right now, some studies at the University of Washington that specialize in water, and we've done 27 runs, something crazy, and we're still doing it. Taking water and looking at the different conditions of the water and looking at a fluorometer, which is investigating how much reflection and refraction goes on in the water. I mean, this is just one example. Something happens. I'll just leave it at that. It's not really answering your question, what's in the water? And the answer is nothing is in the water. There's nothing in the water that we know of other than water molecules. But the water molecules that have gone through compared to the control, there's a big difference. And we've done this many, many, many times. The people at University of Washington are scratching their heads going, what's in the water? You know, what's doing that? So unfortunately, your question is a good one, but right now I don't know what's in the water. I can tell you it's only water, but there's a difference between treated water, informed water, and just water that's a control. That's not necessarily satisfying, but it's compelling if you're looking at, is something happening to the water? Yes. What's happening to the water? Maybe next year?
In chemistry, what happens in the water?
What happens to the water? So are we
Yeah.
looking at changes in bond angle? Are we looking at right now, again, the studies are ongoing. We got a whole bunch, we got labs scratching their heads. What's in the water? We haven't been able to figure it out now. We simply know there's a signature that indicates something is different about the water, whether that's structural, I mean, for example, when I earlier tonight, I said, we've done experiments with treated cotton versus untreated cotton on cancer cells. And if the cancer cells are brought close to the treated cotton, they have a genomic response. Cancer can recognize that. What's in the cotton? Well, I've even taken cotton, and I didn't put this in the talk, I've even taken cotton and put it under an electron microscope, trying to figure out what's in the cotton. It's the same question. What's in the cotton? What's in the water? What's in the recording? And the answer is going to be the same. Something interesting, but I don't know.
And Bill,
What else?
looking at those graphs, it looks like the 300 nanometer wavelength, which is in the ultraviolet range, is
Yep.
where the largest peak is.
Yep.
And as it moves into the visible range, the effect seems to drop off.
Yep.
That's interesting to
Interpretation,
me
John?
because I study the light, you know?
Yeah. So what do you got from that?
Oh, I can't answer you anything. It's just interesting.
Exactly. So that's what I'm saying. Isn't this interesting? You know, I'm not talking about the ultraviolet thing, but you're exactly right. It's interesting. Something's going on, and it's a reliable effect. But what it has to do and how it produces and how does whatever's happening by these measurements relate to genomic changes in cancer cells? How does it change mice with cancer? I'm not here to give you answers. I'm here to screw with your head. Mine's screwed sideways and backwards. And you should hang up as quickly as you can and save yourself, because we're not going to know. But it's interesting. But I have tried even putting a cotton under an electron microscope. There's no changes whatsoever. But the cotton, which you can't see the change, produces, as we'll say, interesting results.
Yeah,
Cancer cells in vitro genomically changing is an interesting result.
and I might not be wasting my time looking at ultraviolet light. That's what this tells me.
Okay, I'm good with that. And all I'd ask of you, John, is figure this out, please.
Working on it.
Next week, we'll have it. What else can water do? Hey, I beat your questioner by one slide. Yeah. What else can water do? So we're doing now, these are ongoing. They're happening as we speak. We're analyzing as we speak. I just ran some data sets yesterday, so close to as we speak. We're doing studies on longevity. So if you take the Restore and you have COVID, that's a good thing to do. If you take the Restore and you've got arthritis, that's a good thing to do. If you take the Restore and you're bipolar, that's a good thing to do. If you take Restore, I don't know why, but what else can water do? If you're a cancerous mouse, it does incredible stuff. We can affect this, we can affect that. I don't know the mechanism, but what else can it do? And so we happened on, I wonder if it has something to do with lifespan. I wonder if lifespan is affected by the water. So now we're doing studies on longevity at Brown. And we're using, as you might expect, worms. So these little squiggly things over here are worms. They're called C. elegans worms. And they're like my mice, which is the standard way that you study cancer. These worms are the standard way that everybody around the world studies longevity. So you putz with these worms and you see if we can jiggle the length of time they live. And they have some interesting properties, but we're not going to have a worm discussion tonight. They have interesting properties, they're reliable properties, there are all these other things, there's simple genetic coding, and they only live a month. And most of them don't make it a month either, so they're similar in longevity to the mice that have been injected, but these worms, no matter what you do to them, including nothing, will die in a month. They'll have reproduced, put themselves on, you're in the next generation a month later. And what you do is, you do whatever you do to the worms, and you see, is there any change in the longevity of the worms? I mean, so that's standard longevity studies. And then you do the analyses, and you find this, you find that, you find the other thing. And what we're finding, just to say in English, the worms are actually putzing around on E. coli, which is what they eat. It's not the E. coli that's dangerous to humans, it's a different form of E. coli. And the E. coli, they hang out, and this is their food source. And what we do is we drop a couple of drops into their water, into their supply. So we just sprinkle a couple of drops, I think eight drops into the food source, and we let the worms see around. We'll see if you're alive tomorrow. We'll see if you're alive the day after that. And then we compare those people who are getting the restore water versus those people who are getting tap water versus those things who are getting no treatment at all versus. And the very short version is this. In 6 replications, because again, I don't believe this stuff, so I got to keep replicating. In 6 replications, we're getting an approximate increase in lifespan of about 10%. 10% to a worm isn't very long. 10% to a human is longer. We don't yet know whether this thing will work also increasing the longevity of humans, but it does on the standard longevity model. We're having a tremendous effect. A 10% change in longevity is not subtle. And it's happening from machine-produced water that's sprinkled onto the food supply. And the question is, what's in that water? What's in that recording? What's in that? I agree. So I'm going down 2 directions, all trying to go increase storability, increase scalability. Recording is has yielded that some good data. Water has yielded some good data. Some of it's really nutty, like the worms and longevity, but by now it's 6 times. I started writing up a paper, the draft of a paper yesterday. So this is hot off the press. We got more experiments to go. We don't have more mice to go because it's getting harder and harder to do mice. Just finished my 19th and 20th mouse experiment at Tokyo University, but it's the mouse research is being shut down. Water research is not being shut down. Worm research is not being shut down. Some things we're allowed to do and some things we're not allowed to do. So researchers are shifting their focus from one thing to the other. And with that, let's take any questions you want to throw at me.
I have a question. How you define informed water?
I define it as water that has been treated, gone through a protocol, and stuck into a device, which I can't talk to you much about because it's proprietary right now. We're looking at what we can do and who we can supply help to people. So it's informed water is water that, metaphorically at least, has been informed initially by a human treater and now is entirely in the hands of a device. Treated water is water that's been treated, hands on or hands in the air, hands at a distance and all that.
Any difference in?
Hey, Alex, there are a number of questions that we have that have come through, so I'd like to
Okay,
get to some of
okay.
these. Bill, you
Hey,
want to stop
John, should I kill the stop share?
Yeah,
Or
you
hit?
can stop your share.
I don't know if I can get back to it, but we'll do it now and see what happens.
All right.
Look,
Well,
there's bodies out there.
there were a lot of questions that were coming up as you were talking. A
Well, I
lot
hope
of
by
them.
now you know I don't have any good answers, but let
Well,
them
so
know.
this is one of the main things I want to ask here. So a lot of them were around the technology used to create the sound files or to do the water treatments mechanically. And I know you're not I actually know a lot about what was done with the sound files because I know who did them. But is this documented somewhere where people, if they want more information, they can go and get the details
Yeah,
of it?
I have on the recordings. I have, you go to my publications, and I think I have 3 or 4 peer-reviewed publications on the recording. I have similar publications on the water, and it's some of it is pretty geeky.
So if you have geeky questions about that, maybe you can find the answers there because it's a little more detailed than what we have time to get into today.
Yeah, I'm giving you the Cliff Notes.
Right.
I'm giving you the general plot, but the details are there. And for the electrical engineers out there, in the materials section of the papers is the materials and the methods and and in the results, you're going to see geeky looking graphs, and you're going to say, what does that mean and why does that work? And I'm going to say, I don't know.
Right.
So give me another one that I won't know the answer to.
Okay. Well, I got another one here. So,
Do you ever have people on who know the answers?
well, they pretend. I don't know.
Yeah, those people scare me. Those people scare me.
Some people do. Well, they know you can answer some things. You can answer like what was done. So, for example, the question came up. You talked about DNA and how you were looking at the genetic changes in the camp. Has anyone looked at the genetic changes in the mice that have been treated? Whether the DNA in the mice is actually changing?
No.
Okay.
We
See,
got
now
shut
you have an
down
answer
before
there. See?
we got shut. Yeah, that's right. We got shut down on that where we could have done it at Brown, where cancerous mice were exposed to the recording, but we got shut down. Yeah, and there's consequences of being shut down. I lost $140,000 on that. Which is not exactly chicken feed. It's not huge, but I did go, ooh, that, and I just finished at Tokyo University just to see if differential cancer models would have differential effects. And we could only take a certain amount because I was down to maybe $150,000. And I shot the wad on that before I could do any genomics. And they were also sick of me because they said, none of this makes any sense. They couldn't disagree that the effect was there, but it didn't make any sense. And if you're in a conventional biology lab, people like to have the illusion they know what they're talking about. I
Yes.
don't feel safer with that illusion.
So with the mice that were healed, the ones that were cured of these things, is there something you can do, like perhaps transfer something from those mice to other mice?
Yes.
Is it like, can you take the blood from one and transfer it to
Yes.
another, and does it have the
Yes.
same effect?
Yes.
So it works almost like a vaccine?
Yes.
That's really interesting.
I have a very large grant proposal out there that has not gotten any bites, that in essence trying to take this phenomenon reproduced in multiple labs, because I'm kind of a fanatic on replication and independent replication. And I have multiple labs that have found taken tissue. If you remember the little bouncing illustration I had, taking from this mouse and giving to that mouse, it looked like a rat. Taking from this rat and giving to that rat, it'll cure that rat,
Wow.
and that rat will be immune for life.
And
What I don't know is whether different types of cancers would also be affected. My suspicion is it wouldn't, because I think it's an immune response, and the immune response tends to be specific.
Is there any work towards a cross-species type of vaccine or treatment? You allowed to talk
No,
about that?
no, I've never taken blood from a mouse and like given it to a person. If that, that you're asking something like that.
Well, I'm asking something like that. It may not
Yeah.
be mouse to may not be blood directly, but something.
Yeah. Well, I mean, that's interesting because the question comes up, what's going to be the delivery system, not mechanism of action, but what's the delivery system? So we can reasonably say the delivery system can come from water that's been treated. The delivery system can come from water that's come out of the gizmo. The delivery system can come from a recording, but they're not all equal to hands-on. So again, we have some. And again, these are in published articles. This is also at Brown. We wanted to see, we were getting the genomic changes in the incubator, playing the recording, and they were startling, and also reliable. But how does that compare to putting your hands around some cancer cells, and doing the crazy stuff that way? And it turns out hands around is stronger than the recording. And that was deduced from it takes longer for the genomic changes to start to occur. And the protein folds are slightly different. So what we have going on right now in Providence is let's look at the old recording. Let's redo the old recording, even though we have many replications, and let's see the genomic changes. And let's look at the new enhanced recording where we putzed around with the signal. So we're taking the Model T and we're saying, what happens if we make the wheels 12 inches instead of 10? Is basically what we're doing.
Mhm.
We're going to have the same problem because the part that's been really juiced in the recording are low frequencies. And so in response to something you asked before, what does it sound like? It sounds like nothing. There's no even background hiss. We got something that our ears can't take, but there are devices that can say something is going on there. So something is going on with the new modified recording in an attempt to juice it up. I'll know a couple of months because I'm going to be pretty fanatical about this and we need replication, 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 replication.
So I know you have this strong affection towards mice. Let's just say.
I used to, and I can't do it anymore.
Well,
You can't
right,
do it.
Mm-hmm. Well,
In order to get the Tokyo lab, just as an indicator here, I spent at least 2 years searching around the world for a lab that would do mice, in a weird way, because this is not conventional biology, and I couldn't find any. And finally, somebody I know contacted me and said, "I think I found you a lab." And it turns out he was in Japan, and it was a Japanese lab at Tokyo University, and he was right. So we used them as a contract lab to do mice because they would allow crazy stuff to happen to mice and let the experiment go longer than anybody like in the States. The States, when I went to Brown to do the mice research, I went there separate trip to make sure they wanted to make sure I wasn't really from PETA. And as I walked around a building which had no markings on it whatsoever, they asked me straightforwardly, "Are you going to throw blood on us?" They've had blood thrown on them. Are you gonna? I said, "No, I'm not. I know this is unorthodox, but we're really doing this research." And they, look me up and down and should we trust this guy, and all of that. It's
well,
that
and,
it's that
and it's
rigid.
ironic because you're trying to save these animals, help
Yeah,
these animals,
but in
not harm
fairness,
them.
I hurt them first.
Right.
Yeah.
Mm-hmm. So, but the goal of this research is the whole scalability and storage is not only related to mice.
No.
I'm guessing the goal is to direct it towards people too.
Yeah, well, we got the clinical study. You know, the COVID trial broke the boundary of what medicine does by a it's not casual. And that was done straight on people, you know, and they had an IRB and they had. So this was conventional kind of research. It's okay to experiment on people as long as they have the ability to say something, and you know, you can't pick on handicapped people. I mean, you're physical handicap, but if you're mentally handicapped, if you're a little kid,
Vulnerable populations.
vulnerable population, and
Yes.
mice are considered vulnerable population. So I was in touch with labs and people trying to work with me from all over the world, and I'm telling you, it was probably closer to 3 years than to 2 before I thought I'd never do it again. And right now, I even went and tried to see if I could do zebrafish. Zebrafish have a developed immune system, and so sometimes you use that for immunological research. But it turns out you can't even use, you can't, I mean, you go down and you fish in the pond, for crying out loud. But you can't do experiments with zebrafish now anymore. At Brown, they have a zebrafish lab, and it took them more than a year to set it up because Brown was protective of the fish. And it's anything with a spine you can't do.
But with people, once you get the consent, and with the studies you've done,
Yeah,
and you see this effect, we were
Yeah.
talking about transferring between mice.
yeah.
Have you considered transferring between people when you see
Yes.
the effect working?
I've considered it, but I haven't done anything yet.
Okay.
Yeah. But that on a long scroll of stuff to do, haven't done it on people.
So as
And
a follow-up,
and again, if we're going to follow the mouse model, we probably have a cancer patient that we treat and treat and treat and treat, and then take a blood test or take a blood sample, you know, and put it into someone else with cancer. That's not going to be as controlled as a mouse, because in the mouse, we know exactly how old they are. They know exactly when they got the disease. They know everything about it. In clinical trials, you don't know any of that stuff. You just know someone has a lump, has a this, has a that. But you know, mice research is much more controllable.
it's much more controllable, but also the clinical trials can help people. And,
Yeah.
you know, and besides the research aspect of it, this is
Yeah.
another aspect that's important here.
Well, I came pretty close to getting a large amount from a philanthropy in New York. And then they backed out. And what I was trying to do is say, look at these data. There may be a vaccine in there someplace. And
And
vaccine, it may be a loaded word that I shouldn't use, but you know, to put it in made up jargon to make it sound more palatable. And
a way to prevent other people from getting the same thing.
maybe, I don't know, and that's another thing on the
Potentially.
scroll.
Mhm.
Can you do the treatment and as prevention? Now, my guess will be, and my guess is no. But because I think healing is response to the need of the healee. If the healee doesn't have a need, what are you doing?
Mhm.
Now, having said that, cotton doesn't have a need, yet it works. You know, every time I open my mouth, I have a little bird on my shoulder that gives me a counter hypothesis and says, "You big dope, you forgot about the cotton, you forgot about this, you forgot about that." So the bottom line is, I'm just telling you, since I've done research here for 120 years, I don't know much. I have lots of stuff that I've done, and I can tell you it's real, reliable, and all that, but really understand. I'm becoming more ignorant with each experiment, which is
Have you-
humbling, wonderfully humbling.
Right, yeah, no kidding. Well, anytime we're truly doing research, it becomes very humbling.
I'm afraid
So
of people who know what they're talking about, because I'm certainly not among them.
have you gone from water to cotton? Like, can you use water to charge the cotton, or vice versa?
I don't know of anyone who's tried it, but these are things to play with.
Hmm.
And what I need is a chargeometer, which
Right.
I have somebody working on that, you know, seriously. I'm calling it, you know, facetiously chargeometer, but you understand how that would open up entire vistas.
If
I
something
knew the
can
cotton
detect
was,
what's in the water for example, or what's in
The
the
cotton
cotton?
is 50% charged. The cotton is 80% charged.
Right.
The water is, wouldn't that be wonderful? Now,
Mhm.
right now, I'm relying on gut, and every once in a while, I rely on sensitives.
Mhm.
I'm not particularly a sensitive, and so I'll exploit anybody who wants to help.
So there's so many other questions that we have that have been coming up, but I want Chuck, you have your hand up, and Alex, we'll get back to you. But Chuck, you haven't asked a question. I'm going to give you a chance to ask.
Okay, thanks. Uh, Bill, you mentioned about some of the DNA changes seen in healed mice or whatever it was. Would it be possible to use gene editing to create the same changes?
Great question. Don't know the answer.
Yeah, that's what I was just
Yeah,
wondering
don't know
about.
the answer. We're looking at some stuff. There are more promising genes than others. So there are genes that are more directly related to immunology. There are genes related to telomere length. There are genes that's what made us do a hopscotch to the worms.
Mhm.
If the telomere length is increasing, maybe, like that. So again, it's like grasping, and who knew a couple of years ago that I'd be playing with worms?
I just, I had one other question too, if I could ask it real quick.
Yeah,
Quick.
When you make Restore, and then one was called Perform, when you manufacture those, why do you label one bottle different than the other? One for Perform versus Restore.
they were made differently.
Well, that's what I'm asking. How were they made differently?
Part of the process of preparation of the water is dilution and succussion, and there are different levels of dilution and succussion that would, and there's also different targets in the gizmo. So we have some tablets that were made in with the help of high energy plasma physicists and tried to capture certain signals. And so, for example, we'd send water and he'd try to capture the water, and then he thought he could infuse it into stuff. And we used that in the gizmo to produce water. So some stuff has come out of human treatment of water and some stuff has not.
And knowing whether it's Perform or Restore, what would guide that?
Our sense and experience about what it's good for, what works. So
Just
I've
from
done, for
experience.
example, before the randomized double-blind control study, I'm leaving out lots of parts. I did a pretest with more people that was less tightly controlled, but included a part where Restore and Recover were separately taken, and then combined taken. The combination of Restore and Recover for viruses that certainly works on COVID. I think it works on flus too, and some variants of the common cold. Those were observation, and tested, but I couldn't quantify one versus the other and all that. That's
Okay.
past my pay grade now. But we need it. We do need what would in essence be a chemistry of this stuff.
Mhm.
I mean, I think I played with dilution. I'm not a homeopath, but I played with dilution and succussion, and something's there. Something's there that makes no sense to me whatsoever. And we're not so nutty that a homeopath starts with some molecules and dilutes them and succusses them, and there's no active molecules left. It's just water. We start with water, but we dilute water. We dilute water with water. Now that'll make your head explode. But it seems to work. And what
I
I'd
have
like
no-
to do, I have a thought that watching some dilution and succussion combinations work better seemingly on plants. Some work better on mammals. Why they do, again, I go, I don't know. But if we could put them all together, and as we're talking about having this chemistry of water, and then the analog would be a chemistry of the recording. That obviously wouldn't be chemistry.
Mhm.
But if we do that, I wonder if we could do combinations of dilution and succussion and produce something more akin to a periodic table. So you'd have reiterating, here are the metals, they're metals at, at this part of the periodic table vertically. What do we have that would be good for mammals? What would be good do we have for plants? What do we have that's good for worms? Do they differ by much? So there's one or two things left to research. Three,
Thank-
maybe. Once
thank
we
you,
get to
Chuck.
three, we're done. Yeah, we'll know everything.
So Alex, I'll let you jump in with a quick question if you
Okay.
want.
Yeah, you mentioned a few times about signal. Is that the electromagnetic wave signal? And what is the source of the signal?
You ask annoyingly good questions. Yeah, we're doing electromagnetic signals and readings. The source of it, I mean, I can't tell you really what the source is, but I can tell you the procedure. If we go into a Faraday cage and get rid of extraneous signals, and we take a piece of cotton and we do the healing stuff, which again, I can't explain here, but it's on the CDs. You can get it really cheap. There's a lot of different ways to learn it. Play with it yourself, see what you come up with. So if we put people in there with these detectors, we get really interesting results. If we do, I've done informal stuff, but it indicates that it's a path worth pursuing. We've taken a lot of people cycling, and that's my healing shtick. We've taken a lot of people together cycling, and by a lot of people, I'm talking, like pushing 300 people, and had the 300 people communally cycling, running their phones and recording it. And that seems to produce stuff. I've gone into an audio studio, your basic sound studio, with two mics opened up wide. And we stand there waving around, doing our shtick. That's a different kind of a signal. And we've taken it into the lab and done genomics on it, and that produces genomic changes. It's not as strong as the Faraday cage, which isn't as strong as the hands-on healing, but it shows there's potential here. And so if you ask the most obvious question, what different ways can you record healing intention? My answer is going to be, hell, I don't know. The Faraday cage is not sacred. It's a packet of technical potential that we're playing with now to see if we can make it stronger. But what other ways could you do this? I don't know. And if we're down at 3.5 to 5 hertz.
Out of everything that was recorded, you don't know which one was the active component of it.
No.
So there was a lot of things that were recorded, and they were put through a transform of sorts, and that transform produced a recording. That's the details that are in the paper that are beyond.
We don't want to talk about that here.
Details of it.
Yeah, you can go down a geek road into a black hole, and I invite you to do it with gusto. And if you find flaws, I welcome your flaws.
Yeah, because we want to learn more about it as well.
We want to learn. Yeah, I'm not suggesting I have the way to record. I mean, give me a break. I have all the materials that you can store. Give me a break. I'll show you what I've done. And, I've done a bit more than I'm letting on here, but I've done what I mean, it doesn't even make sense to talk about a fraction of what we could do.
Mm-hmm. So
there's
thank
plenty
you,
of
Alex.
work. There's plenty of work.
So Bill, I have one more question, if you can bear with me just one more moment.
Would you repeat that?
And it's not an easy question, but within our field, we often discuss experimenter side.
Yep.
And the fact that the experimenter, the people who are doing the experiments, can have an influence on the results of an experiment. And this is not only in parapsychology, it's in pretty much across the board in all the fields.
Without doubt.
When you see such consistent and such strong effects in your research that you're doing, how can you protect or dismiss the idea that experimenter side might be responsible for the effects.
No, I can't. I can't at all. I am a proponent of not proponent, but I completely acknowledge experimenter effects. I remember being with Jacques Benveniste, if we're talking about playing with water. I worked a little bit with him in Paris, and we were out having lunch in Paris a couple months before he had the unfortunate operation and death. And he had just discovered, to his horror, that the memory of water depended on who was in the lab. And it wasn't like they had to be working on the experiment, they just had to be inside the lab. And Jacques was flopping on the floor, angry, because he wanted a straight, materialistic. And I said, Jacques, this is more interesting. He didn't find that humorous at all.
But that does seem like something you can vary to see if you get the same results, or see if you can get more information about who might be responsible, or whether there's a combination of people responsible.
Yeah. So we've done the recording, for example, with 3 people, because I had access to 3 people. We did cotton because we've done experiments on cotton, and we have a clinical history as well as an experimental history. I've done. But if you remember in all the little bullet points I had, we don't know the effect of multiple healers. Does the 3 people, do they produce half the effect size of 6? I don't think so. But you know, what happens when you combine, and does it matter who's in the cage? Does it matter who's in the sound studio? I wouldn't in any way, shape, or form minimize that. I just, if I can store it and scale it, and that stored is independent of anybody, any one person or 2 people, and it appears to be, then that doesn't even minimize the experimenter effect. The experimenter effect can have a lasting, real change, which conforms to more conventional kinds of analyses. But the idea of experimenter effect, I think you'd have to be nuts to deny it.
So there are ways that you could, like, if there's experiments that are run that you're not involved with,
Yeah,
and you're not aware of things like
Yeah.
this, then you would not, people wouldn't, it would be less likely people would look at you as an influencer on those sorts of experiments.
Yeah.
And I know you've changed up personnel and changed up timings and things like this to blind yourself
Yep.
to the results of experiments. But what I'm hearing you saying is, hey, if it's an experimenter effect, let's take advantage of that.
Yeah, use it. Beautiful. It's like,
Yes.
it's like I have I'm making this up, but I found something that's as good as a placebo. Wonderful.
Right.
Yeah, that's good. Oh, it's just a placebo. What are you talking? Just a placebo? You know, it's yeah. Placebo research is seriously interesting, and I think most people doing it are going up the wrong tree. But another night.
Bill, we could talk, well, and we have talked for hours about these sorts of things.
Yeah.
But I appreciate you taking the time with us tonight. And I appreciate all the patience with everyone trying to go through the questions and trying to work through this. I definitely encourage you to go to Bill's website, bengstonresearch.com. Is that right, Bill?
Yep.
Go to
One word,
Bengston
Bengston Research.
Yes, look up Bengston Research, and you can learn more about everything that all the things that you're curious about. Bill has also done a lot of interviews. They're all over the internet. You can find him speaking on lots of different topics, including going into detail about his methodology. So
Yeah.
I definitely recommend you explore that in more detail. Thank you very much, Bill. And thanks everyone for being here.